Okay, hi guys. Yes, sorry, I'm at home with COVID, and but I didn't want you guys to fall behind because we don't have too much time for this unit, and I really want you guys to gain the experience. So just remember, you need to refer to the presentation if you need help, unless you are lucky enough to have a reliever that has microbiology experience, but it's not their job to facilitate the practical. Okay, so please try and work as independently and help each other. But luckily, you've done all these techniques before. So as you can see on our screen here, I'm hoping that your two plates that you did with the streaking out method from last week gave you something similar to this. Um, this one is obviously from E. coli or a similar bacterium. You can see there's no fuzziness, and the fuzziness usually refers to a fungus. Like if we go up here a few slides, you can see that one over there, or that one that the red circle is around, or this one. Yes, thank you. I forgot I put an animation in here. But if it's fuzzy like this one or fuzzy like that one, you grab some where the color changes, and that's where your spores just begin to release. Don't go all the way to the middle because there's too many spores in there. And then you either take an inoculation loop, or you take just a straight needle and you get a little piece. Now, you need to be able to see it, okay? And then you tease it out on a microscope slide, not like this, but on a microscope slide. So you take either one loop and one needle, or however you can, but it needs to be spread apart. Alrighty. So when we then, and I want you to take from a section where you can hopefully pick up a single colony. And remember, this is only for your fungal petri dish, not for the bacterial, because for the bacteria you need 1,000 times magnification, and we do not have that facility at school. That's usually when you get to university level. Okay, let me just jump to a different presentation now. Hello, and please. I'm using 3 different presentations to try and make sure that you know what to do. So you'll remember that you take your sample, you put a droplet of iodine stain on your plate, and then you put the sample inside that droplet of iodine stain. Then you tease it out, and then using that 45-degree method I taught you, you slowly drop it onto it, and then you use a tissue paper to remove the excess. Okay, and then you're ready to go and investigate it under the microscope. Now, what are we looking for? Because in the due work, I asked you to look for fruiting bodies. Now, fruiting bodies of fungi are in here. So here I've got the same thing. You're going to have to grab from this area of isolated colonies, and we're looking for these types of structures. Okay, they're all you're going to have to go to at least on 40 times, or then 4 times, 4 times 10 times. You will not see the detail. You're going to have to go up to 100 times or 10 times 10 times. If you're really lucky, you can go up to 400 times. That's the biggest you can go. But for now, I think you'll be okay. So there's 3 of them that look very much similar, and I'll go through here. So let's look first at your petri dish. And you might see that if it is Rhizopus, then your petri dish will be a blackish grayish color. Okay, and important, if you find we call this thing a sporangiophore. If you find a encapsulated one, you'll see that it looks really sealed. It doesn't have rough edges. The spores are not really sitting on the outside. You can see it's smooth. If you look at this area here, you'll see there's a membrane around it. I always refer to it as a hairnet. Now, Rhizopus and Mucor both have this smooth edge, like a hairnet, that they have over them. The difference between Rhizopus and Mucor, which I will show you next, is these things called rhizoids. So if you see something that has that, and then like a root structure under it, that is the rhizoids, then you know you've got Rhizopus. If you have one that has its hairnet on, and you can see there the hairnet, see the hairnet, okay? So it's not outside. Then you can again see the hairnet there. Then, but there's no roots, then you know you've got Mucor or Mucoralis. Okay, so Mucor and Mucor, the most likely ones you will find from here is going to be most likely Aspergillus or then Penicillium and potentially Mucor. Okay, so I'm going to skip over most of the other, but Penicillium is usually going to be a greenish color. You can see there's different, and remember this is the one that Fleming identified penicillin from. But can you see it's this little hand type structure with spores in a line above it. Okay, so you're looking for something like that, little fingers, and then the spores come off the fingers. It can sometimes be this flower arrangement, which is really pretty, but it's not as round as what you would expect from Mucor and Rhizopus. Okay, so this is the kind of arrangement for Penicillium. And then Aspergillus, this is the one that you're most likely to have. It looks, if you quickly go back to over here, you can see that it looks quite similar to Rhizopus and quite similar to Mucoralis, but Mucoralis and Rhizopus got the hairnet or the membrane, whereas Aspergillus definitely does not. And that's your biggest distinguishing factor. You'll see there's obviously no rhizoids, and if you do have Aspergillus niger, then it's going to be the black fungus. Otherwise, it can be green. Most likely the one I isolated and looked under the microscope last week looked very much like this. Actually, it looked more like this. And I will show you a photo in a short while. Anyways, so these are the ones that I suspect you will have. And the presentation that you're going to open for that is this one called Microscopy, Isolating Pure Fungi Culture from Soil. And when you open it, you will have towards the end, after we discuss all the techniques, and I'll go through all of this with you at some point, but you'll have the keys over here for you. There are others like Alternaria, Fusarium, Verticillium, Rhizoctonia, and Pythium, and so on, that you will have a look at. But in general, as I say, look, Aspergillus, it doesn't have the hair net on. Rhizopus, it's got the hair net on. Penicillium, it's got the little fingers. Okay, so try and identify it, then take a photo with your phone on the microscope, and please upload it to do work. Alrighty, just have fun and try and get a nice picture, but work fast because there's not a lot of time. Okay, thank you guys, have a great day, bye.